We've got some people coming in, and the time is now the time to start. So I think we're gonna officially start this. So we're here with Dr. Robert Arking, who does drosophila research at Wayne State University, and he's graciously decided to spend some time with us to tell us about his research today. So I'm just going to let you introduce yourself and take it away.
Okay. Well, thank you. Thank Aaron for inviting me to give a talk to the forum. It's always a pleasure to talk about my favorite topic, which is biology of aging, and especially to students who are interested in this for one reason or another. So that's good. And we'll see if we can add a little more understanding to Aaron's comment about how mysterious this whole thing is. Okay. We all know that fundamental biological processes are highly conserved across phyla and species. And so it's only logical to ask if pro-longevity processes known to be active in flies may also be active in humans. And the answer, well, will come at the end of this talk then, okay? Now, so let's step ahead. What's the first goal of modern geroscience? Today, people die about 78 or so, and they're sickly. So they die of some kind of age-related disease. And that's not very helpful. It's not pleasant at all. So what we'd want to do is to have people live to more than 100, let's say between 100 and 110, and die healthy. And that's, people may say that's a problem. Why don't we try and live for 1,000 years? I would suggest we take the first small step first and see what we can do. If we can do 30 years, then you can start thinking about 100 years or 1,000 years or whatever. But first, let's see what we can do in this one. So I was just telling, and so, okay, enable people to die healthy at an advanced age. Okay. I want to admit, this is some people that we've collaborated with. It's an incomplete list, but there's a lot of people. And the whole point of this is simply to show that, even though I'm going to talk about data came out of my lab, there are a lot of people that did this work with me. Some of them had skills that I didn't have at all, of course. And so it was a collaborative affair, just like much of modern science is. I think if you look at the top, Michael Rose was probably the person who first had the idea to use flies. He was a mathematically inclined evolutionist, did a postdoc back in 1975 or 6 with Brian Charlesworth in London, and did some of the early work on that. When I came to Wayne State in 1975, I met Leo Luckenbill, who had been hired a year or two before me. He was an ecologist, but he had a deep interest in evolution. And I was hired, as I mentioned, to do, a year later, came in to do developmental biology, but I was always interested in the other end of that, the life cycle. What makes a people, why should an organism die? If it can create itself in the beginning, why can't it just repair itself at the end? And so Leo and I were friends and talked at lunch and so forth. And we decided the flies would really be a good way to do this. They're cheap, they're good genetics, you get data relatively fast and so forth. Leo knew about Michael's work. He invited him down for a seminar, gave a seminar. We sat around for a weekend talking. And at the end of the week, we decided to form 2 teams. Leo and I would work together and collect wild flies and subject them to selection. I'll tell you what that's about in a bit. And Michael would use an existing line of strain. He would use large backgrounds. He would probably use 1,000 flies per generation. We were going to use 100 flies per generation. So there are some minor differences, but I think Michael was the first one to start it. Leo and I picked it up and went after it. So it is a collaborative effort. Okay, here's a chart of the first experiment, well, the main experiment. This is 20, it's a little fuzzy, I'm sorry, but the original papers were fuzzy too. This is, whoops, 25 generations across the bottom. And then this is lifespan, and we've exaggerated this leg to emphasize the experiment. Okay, we're looking at the RA is the name that we gave, the code name we gave for normal lived flies. LA is one for long lived flies. And so in this case, the dotted lines here are the LA flies. And so one that we started one that had 54 days, another that had something like 40 days or something. And they just sort of ran up and down and didn't go past this. And the mean lifespan was 46 days. The ones that we said to be long-lived, and the mechanism we used for selection was to take generate the next generation from eggs that were laid by the oldest females in the population. So that means it's a little difficult. You always have to have backups and so forth. You don't know if this gal is going to die tomorrow or not. Should you collect her eggs? All that kind of stuff. We started that. And so from generation 1 to 5, it was not much change. And then in generation 5, all hell broke loose. A lot of the flies died early. There were some flies, but they're laying a few eggs, and then they laid a few more. And then those eggs, when we put them into the 6th generation, took off, and you can see they just went up like a rocket. So clearly something happened back here. Probably the key genetic changes that I'm going to talk about later happened in this case. We have no evidence, but that seems reasonable. May not be right, but it's logical. And so those things went up. One other point, I said we rose them under 2 different conditions. One was, well, several conditions. One with the LAs were with high density, so there were a lot of eggs. So there's a lot of larval eggs around there. And the other in low density. You notice that the ones laid under low density, repeatedly, other data showed this. They don't go up to as high a level. The ones that were laid under high density, some stress, lived a long time. The important point here is that's a social effect that affects longevity. And this social effect is found in all organisms, and it becomes more and more important as a species becomes more and more social. The most social species on this planet is, in fact, the humans. And you can make a very good case that 75% of the variance in mean longevity between any 2 members of the population has to do with a social effect. It's not just your genes that do this. In fact, it's the social effect that alters the expression of your genes. And if you try only to control the gene pathways and do nothing to control the social effects, you will not succeed in making people live longer. So and I could talk for this for a long time. A lot of other people have been to work, but that's very clear. Okay, so we have these long-lived animals, and they had a doubling. How did that come about? This is the LA animals are done in replicate, of course, and all these things are done in replicate and repeated everything lots of times just to make sure. And so what we have here, these are the long-lived, the RA, the short-lived flies, and they are living pretty good. And then about 28 days or so, you see some deaths start to occur in both replicates, and they start to come down. So the end of the health span is this blue thing here. This is the health span for these normal-lived flies. And then they go down this senescent thing, and they are losing, we have data, I'm not going to report it, that they're losing different functions as they go down here. And they lose it at about the same time. So this is their senescence span as they're falling apart. And then the maximum life was about 70 days here or so for the longest lived animal. And the median lifespan would have been about 40-some days here. The long-lived animals live a lot longer. There are some deaths that occur in here because you can see the curve starting to go down a bit, but then they come down just as this does. And so in fact, we have an extension of the health span. Instead of being only 30 days long, it's 60 days long. And the senescence span is the same thing, about another 30 days or so. We have found, and I'm not going to present this data, but if we give different drugs to the senescence span at different times, we can extend this senescence span as well. Although it doesn't seem smart to expand the senescence span when you have sick animals. So you really want to increase the health span over here. So what we have is they've doubled the health span. This is a real increase in longevity. We were always afraid that you get an increase in senescence and not in health, but actually it turned out to be the right way. So there's a health span and a senescence span, the two parts, and both of these are somewhat plastic. So now this is sort of laying out what I'm going to talk about in the next time. We're going to compare the RA and LA animals to identify what we found the 7 physiological and genetic changes that led to the extended longevity. The first one is resistance to reactive oxygen species, ROS. These are highly reactive molecules which cause tissue damage. The second is changes in the mitochondrial efficiency, which gives rise to changes in energy allocation. Those are, I think, the 2 important things. But we're going to see that Exercise and gene expression is different in the LA animals, or mesis, which has to do with activating various repair mechanisms, is different. The dietary restriction response is different. There are changes in fecundity, and that led us, all these data taken together, led us to do the DNA sequencing of the LA genome, which gave rise to a new proto-longevity pathway. And then we're going to take that data at the end and compare it to human centenarians and see if, in fact, some of the data are overlapping.
May I ask a question, Robert?
Sure.
Can you go to the previous slide?
Yes, if I can get there.
So, no, the reactive oxygen species is what I
right.
wanted to ask about. How do you measure resistance to reactive oxygen species?
That'll be the next slide.
Okay.
Okay. And if it's not, tell me. Okay, so here's a comparison of LA flies and a summary of the comparison of LA and RA flies. RA flies are on the bottom. They're measured at 1, whatever that value actually is. And so up here, for example, and the LAs are measured in response to that. So longevity is 40% longer in the LA flies. The lipid content is 2/3. It's down by 1/3 from the LAs. And the glycogen content is very high, about 80% higher. So these animals are very different, just right offhand, just in their metabolism, they're very different. The protein content is about the same. Paraquat resistance, which is a measure of ROS control, is more than twice as resistant here, and I'll explain those enzymes. And actually, it's a charge after this. Desiccation, water balance is about the same. Metabolic rate is exactly the same, but the metabolism itself is very different. We couldn't get a numerical thing, but the example is that the pentose shunt is used very high here, and it's not used at all in the other animals. So these animals are different, and metabolic rate's the same, but the metabolism is very different. And I'll show you how that comes up.
What that is? The pentose shunt? I've never heard that term before.
Oh, this is pentose shunt, where you generate not NAD, but NADH,
Okay.
and NADPH. And so this is an alternative way, and it uses, it's a different, I knew this once when I was doing organic.
Yeah, it's an alternate metabolic pathway. I see.
Right. Now let's talk about the ROS. It'll be this charge here. The oxidative stress resistance is about 60% higher in the LA animals, and that is due to different enzymes that come up. One is copper zinc sod. This is the activity of the enzyme is 50% higher. The copper zinc side is the enzyme that detoxifies the ROS, and this is the one that's outside the cell. And there's another one, the manganese side, that's inside the cell. So the cell, there's some protection on in and out. So there you have like a 50% increase in enzyme activity, twice as much enzyme here as in the RA, I mean, and 3.5 times as much mRNA from the copper zinc side. On the M side, you have 4.6 times as much. So there really is a lot of this. This is inside the cell, and this is really important. And then catalase, which is another enzyme that can, it's about 2.5 times relative to the RA. Genes that have nothing to do with enzyme, with the oxidative stress resistance, for example, alcohol dehydrogenase or things like that, there's no difference at all. They are the same. And you can see that the oxidative damage in terms of lipid and protein are significantly smaller in the LA animals than they are in the RA and the regular animals.
And how does one measure that damage?
I've forgotten the exact protocol, but it was in fact smushing up the flies and taking extracts and measuring it. This paper came out in between 1987 and 1995. I don't remember exactly, but
Okay.
if you're interested, if you drop me an email, I will
I'm
send
very
it.
interested. Yep.
Okay. So we have an animal that has a much better oxidative stress response. And by the way, I don't have enough time, but we can do it in the questions. I have a cartoon that shows how these things interact inside the cell. And we can take a look at it then if you're interested in it. The flies do not have a low metabolic rate. There was a theory that you lived long if you were very parsimonious with your ATPs and you didn't spend them rapidly. So you'd live a long time So you'd have a long life, but it seems like it'd be a very boring life if you can't do anything with it. And we did this experiment. These are two slightly different experiments and different changes, but they come out with the same data. These things overlap. The LA and RA data, do overlap for most of the time. When you do the stats on these, of course, you can only do it for this period of time when they overlap. Then, in fact, there's no statistical difference between these. So they have the same metabolic rate. They do not have a lower metabolic rate. And so if that might suggest, without any data, that might suggest that there's do they have a more efficient metabolism? And that's a possibility. In fact, it takes us to the next point, that the long-lived flies have lower levels of mitochondrial hydrogen peroxide production in vitro.
Mm-hmm.
And this was done some years ago. And you can see at the beginning, so that at 15 days, the LA is a little bit lower than the RA, but it's certainly not significant. And then as you move ahead, so this is about the 28 or something, these are starting to separate, and the RAs start to increase. They both initially the same slope, and then the RAs take off like a rocket, and they increase. And of course, there's no RA data here because we're all dead by this point.
Yeah.
And the LAs are going up more or less at this point.
So the design is more resistant to degrading in such a way that it produces a lot more peroxide in this case.
Yes. And so actually the RA is wasting in effect a lot of ATPs because it's generating hydrogen peroxide, which is damaging cells. And so you then have to use energy to repair those cells. And if you don't repair them, you just go down the tube because you're becoming more and more less efficient. And so if you take over the lifespan, there's about a 20% overall difference in that the LA is about 20% more efficient in terms of than the RAs, in terms of not wasting ATPs on hydrogen peroxide. But this is important. Now, so back in the day, some years ago, we decided to take mitochondria from the RA animal and put them into the LA animal. We didn't have CRISPR at that point. We did this by making crosses. It took it took about 6 or 8 months to generate the stocks and all that, so forth. So what we have over here, let's start up here. So this is an LA male. The first LA refers to the source of the nuclear genome, which is LA. And this is an LA mitochondrial genome. And this animal lives 77.1 days plus or minus on a particular diet. If we then take and put the RA mitochondria into an LA animal, RA mitochondria into an LA animal, we now get a lifespan of about 64. That's significantly different. And what that does is rescues, if you put the RA mitochondria into an LA animal, you rescue the RA longevity. And the same thing happens over here if you change the diet a bit. You get the same result. Now, if you go to the other one, here's an RA male, and lives 67 days. And now you put an LA mitochondria into an RA male, you now get a lifespan of 72, which is definitely different. And so, and you get the same thing over here. It wasn't significantly different, but it was in the right direction. If we had more animals, maybe it would have done better here, whatever. But the point is that in both these cases, the mitochondria affects the phenotype. It determines what kind of phenotype that animal is going to have. And that's very interesting. Same thing happens in the females. And you go down there, there's one case here where it just didn't got no effect, and that could have been for any number of reasons. But in 7 of the 8 cases here, there was either a significant change or at least a change in the right direction. So the heterologous mitochondria rescue the longevity phenotype of their source genotype. That's interesting. Okay, let's see. Let's go here. Now, the energy is also reallocated. Once you change the efficiency and you have the RA and LA animals have different levels of energy floating around, now you can get, and you can reallocate them differently. Energy can be reallocated for reproduction, which is the prime thing for all animals. If you don't reproduce, then you go extinct. So reproduction is a major source, because that's first priority for reproduction, for use of all resources. And then what's left over can go for maintenance. So the RA animal, both animals here have the same amount of energy. This is in microliters of oxygen per day per female, yes. And this is like, this was a fine, Vasily Novoselic came to visit and worked in our lab for a couple months from Moscow. Excellent mathematician and interested in this kind of thing. Did a great job. But they have the RA and LA have roughly the same amount of energy available. And this animal puts 45% of its energy into reproduction, and what's left goes to maintenance. Now, the other one puts about 35% of its into that. It's actually somewhat less than this. I'm never quite sure why that happened, but it puts a lot more into maintenance. And so this animal lives longer in part because it can do maintenance of structures which are repaired. It just repairs them. Okay, now the next one, right.
This is measured per individual, not per time span, though, isn't it?
It's measured on individual flies. So we knew what the all the data was for the flies and how many eggs everyone did and how much energy they used during the time period that we measured them. But it's on individual flies.
Because the generations are longer with the RA flies,
yes.
there are fewer total flies reproduced during that time span than compared to the LA flies.
Actually, there's more, but there's more and there's also less. There's a little, this is an interesting, you put your finger on an interesting point. And I think it would be the next slide or no, another slide, but the fecundity issue is interesting, and I'll come back to that. Another thing that changes exercise, the ability of the animals. If you look at the RA animals, if you donate exercise, I should say, there exists a fly treadmill that you can exercise flies on.
Okay, yeah.
And this is done by a colleague of mine, it was invented by a colleague of mine at Wayne State, Robert Wessels. And the first time I saw it, I burst out laughing because it was so funny. These flies are running up and down. But in fact, it's a very active thing and it's a good deal. It's just the first thing, most people start laughing when they first see it. Let's go back. I came up on it. So if you have an RA animal, then it has a certain shortest lifespan on this chart with no exercise. If you put them on the treadmill, they actually will live a bit longer, and significantly. It doesn't look big here, but it is statistically significant. If you do the LA flies and no exercise, they have the longest lifespan. And if you exercise them make them go on the treadmill, you actually shorten their lifespan. Now this happens
You're
because
using up the energy, and it's very well allocated due to your evolutionary pressure. So when you're forcing it to reallocate it, it's less efficient.
That's part of it. The other part is that it became obvious to us early in life that we didn't have to look at the labels on the bottles to know if this was an L animal or an R animal. That in fact, just looking at the movement of the animals, the LA animals always moved very rapidly. They just moved all the time. Even if they were the same age as the R animals are sitting around on the sort of like rocking on the porch, and the other guys are running around the yard. And they are so active that we believe that they're constitutively exercised. And we think, no data on this, but we think that then if they're essentially exercised to the maximum, then you force them on the treadmill, you're essentially overstressing them, and they just can't. It's over the hill. That may be a reasonable thing. I will point out. So here we have long-lived animals that can run. Okay, it turns out in a separate experiment done by people some years ago at the University of Michigan, they selected rats to run fast, and the fastest running rats lived longer. So being highly active and being long life are something that travels together, at least rats and flies. So there's some kind of an interesting connection there. Exercise. There are, as we did some data, there's about 550 genes that are brought up by exercise. or by genetics. So these are the genes by 550 in exercise that are found in the LA animals, and then just in exercise on the other animals. And normal exercise only brings up at most 80% of the genes involved. So it's enough sufficient to let us live a normal, healthy lifespan, but not sufficient to let us live an extended lifespan, such as being a centenarian. And the other thing that's interesting, these are some of the genes representative that were up and down regulated. And when you look at them, they're all essentially metabolism genes. There's no genes at all that come from the insulin-like signaling pathway, which is the basic pathway thought to generally regulate aging. So just file that fact and we'll come back to that. There is this other point, hormesis refers to the fact that a mild stress can induce a repair mechanism, a repair process in the organism to relieve that stress. One of the classic things is giving a heat shock to a young adult. In this case, we took young animals and put them at some higher temp for an hour or so, something like that. And we got an extended lifespan. You did it young, but the effects lasted their whole lifespan, so that they lived much longer, even way out into the senescent span. And that happens because the heat shock proteins are activated. These are chaperones that involved in protein folding and so forth and so on. A lot of processes that they monitor. When you do the same thing to give a heat shock to an LA animal, it has no effect on lifespan. And that turns out to be because our interpretation was that the heat shock proteins were constitutively active in the LA animal. And it turns out we're just getting, pardon me?
They're already on.
They're already on. And that was verified. We were just ready to do that when somebody in Michael's lab verified that. So we didn't do the experiment because we're running parallel. But yes, they're always on. And if this is on, you can also make the assumption that other similar kinds of stress pathways would be on. And there's a whole lot of stress pathways that we can talk about, but not now. Okay, so let's see. Okay, the LA strain does not express a dietary restriction response. Dietary restriction, restricting the diet and finding out that this would increase longevity, was the first intervention discovered in 1935 that could extend longevity. So it's a classic thing. And what we mean is something over here. If you take flies, the LA animals are these blue lines. If you give them small amounts of food, one is a normal amount, and then these are portions to that. If you give them not too much food, they don't live quite as long because they're undernourished, obviously. Then they come to some point where it's a peak, and then as you increase the thing, they live shorter as you give them more and more food. And if you take it up to like tenfold, it's like force feeding the animal, they really don't live very long, but they're big and fat. So this is the peak value. It's a DR response, as it's called. Now this is the LA animal, and in fact, it reaches this point, which is maximum, and it stays. That's a straight line. There's no significant difference between these 3 points. And so it becomes a straight line. There's no dietary restriction response. That in and of itself is not important, but it's suggesting that there's a big metabolic change of some sort that's bringing this about. And that's what the real point is about. Let's see. There is an evolutionary problem posed by the LA fly's high reproductive activity. When we first measured the number of eggs that the LA fly lays, we were really surprised. We expected it to have fewer eggs than the RA. But in fact, it came to be a large number. Lifetime for number of animals we looked at was 1,437 for the LAs versus 1,030 for the Rs. That was per female per lifespan, right? And that rose a real question. How could a LA female have the same metabolism, have greater longevity, higher fecundity than the RA female, and yet not be found in the wild, only be found in the lab? And why, in this case, isn't this animal, why, if the LA is so damn good, why couldn't we find it out in the peach orchards? And it turns out that this data is true, but misleading. Let's go and see what we're going to do about that. Whoops. So when we looked at these data on the left, there's the RA and LA. We looked at survival, egg to adult survival. And 84% of the eggs from the RAs hatch and give adults, and only 68% of those from the LAs hatch. That's a 19% difference. That's huge. If competing animals in a normal evolutionary scheme out in the wild, if they have a 1 or 2% edge on somebody else, they can make a very nice living on that. Thank you very much. 19% is atrocious. It's huge. We said we did something wrong. So we went ahead, back to another orchard, got more flies, did the whole damn thing over again. Like 2 years later, whatever it was, got those experiments. And so here's the new data. 88% overall egg to survival for the RAs over here. Oops. Or RPH as we call them, sorry. 88% here, 78, 72%, 11 to 14, same range as this, 15% if you're taking even these across here. This is huge. And the reason that the LA flies are not found in the wild is that they're outproduced, reproduced, I should say, by the RA flies. And so the flies that are really adopted to live in the wild are the RAs. The LAs are unusual and in a very odd sense, weak.
Well, you have a very strong artificial evolutionary selector to get them, right?
That's true. That's true. We're focusing on one thing, and perhaps, I suppose, if we had focused on several factors at once, it would be a complicated selection thing. We could get a different answer, but we didn't do it. But you're right. That's a good point. Okay.
But evolutionary success, as far as I've understood it, is a relatively low bar to reach. It very much favors quantity over quality. And these flies in every other respect seem to be better in every way.
They are better in every way, except for this egg to adult activity. But yes, they are better. If this though is a general phenomena, then in the future, will very long-lived humans not be able to reproduce sufficient offspring? I just asked the question, throw it out. Do you want to create a group of people that can live forever and not reproduce?
Well, I wouldn't say forever. Longer, maybe.
Yep.
Yeah.
Okay. Let's go this way. There's a sensation that this might be true. There is at least one report that lower fecundity is found among human centenarians. That may not be the question I asked about humans not reproducing. Long-lived humans not reproducing may be more than just a theoretical question. And this is a correlation coefficients, a correlation chart of some of our charts, and you can see there's a significant difference there. The difference between the trade-off between longevity and developmental viability. You can have one or the other, but not both. And that's in effect, the fundamental rule of the universe is that there is no such thing as a free lunch. Everything has a cost. And you pick the cost you're willing to pay. Okay. But the animals, by the way, die in the pupal stage. And the reason is simply not known. We never got around to doing that. Now, this is the conservation of the insulin-like longevity mechanisms that are found in the in organisms from yeast to mouse to humans, I should say. And a lot of effort has been posted on these, and people do mutations in these various parts of the pathway, and you can increase or decrease the longevity of the animals. And a lot of effort is focused on these insulin-like signaling systems. And so we decided to take some mutations of the insulin receptor and the IRS and put those into our flies and see if they would, what would happen. Whoops, this way. So this is, and again, we did these by crossing them in, not by any other way. This is the LALA, lifespan for this control. And then this is a case of an INR, and this is an IRS mutation here. And they give us these two lines, which do show a reduction relative to the LA, but not a very big one. But when we put in a mutation for the PI3K, we got a huge reduction. And we interpreted this to suggest, not prove, that the insulin-like signaling method pathway is probably not responsible for the extended longevity of the flies, because you got a very small response from IRS and INR, which are unique to that pathway. The PI3K is in lots of different pathways, including the insulin-like signaling pathway, and yet it caused a big decrease in lifespan. which suggests that maybe it's another pathway someplace that's in which that is involved that is driving the extended longevity, and it's not the insulin-like signaling pathway. So this made it important. This made it obvious to us. We had to go ahead and sequence the genome and find out what's going on. And this took a couple of years to get the money, and a couple of years to stand in line and get a. Everybody was at this time. Everybody was lined up at the DNA places trying to get their stuff sequenced. Eventually, we got it out, and this was published in 2018, end of 2018. The top line, this is flies from our lab, and it was done by Fabian. These are two grad students from Thomas Flatt's lab. This set of genes over here is from Michael Rose's lab, and this is from the Remolina lab, which took our did a replication of our experiment and did it exactly the same way. So we have three independent sets of long-lived animals generated by the same process. And all three of these are enriched in TOL and IMD genes, which regulate the innate humoral response. There was no effect, no increase or decrease, or no alteration of genes in the insulin-like pathway. So we regard these immune pathways as arising from the evolutionary-based selection regime common to all three lines. They are public pro-longevity signal pathways, which are not activated by mutational-based experimental methods. And I suggest that the answer here is that your method determines your answer. And if you focus, and mutational data on insulin signaling that's in the literature is quite right, and you can alter it quite often. And it involves TOR and all the rest, but you can alter the longevity. But there's other pathways. And this one suggested it's the insulin signaling, the mutation, immune system. Now let's look at this. This black and the gray and black thing is a part of a summary chart I use in my class. All the different known signaling pathways that affect longevity. So this thing here would be the insulin signaling pathway. Over here is a group of pathways related, TOR, of course, but this is also involved where mitochondria come in and resveratrol. There's a whole bunch of things that take place here, and protein translation. There's, of course, stem cells, gonad stem cells can get involved in extended longevity. Environmental signals that give socially based longevity, working clearly through CNS and hypothalamus and so forth and so on. We don't know what happens here. I put this, we don't know how this fits in to the existing pathways. So I just stuck it off on the side of the chart. We'll figure out, Thomas and his colleagues are trying to figure out where this is. When you turn on, when you activate TOL, you shut down the part of the immune system that's involved in immune inflammation. And when that happens, you get extended longevity.
Yep.
If you activate cactus, which turns on the inflammatory, you get a decreased longevity. The interesting thing then is when you shut it down, you've shut down the inflammation, but you have not shut down the ability of the animal to produce specific proteins that can kill bacteria or fungi that pathogenic bacteria or fungi that try to infect the animal. And so the animal stays healthy because it has the killer cell, so to speak, part of the thing working. But the immune system, the inflammation is down low. Okay, now, so that's where we are at this point. And we're still, and Thomas and his people are working out exactly how this mechanism is working, where it fits in and so forth. And that'll be coming out whenever, I don't know.
I have a question about the last bit. Could inflammation have an impact when inflammation is turned on? Could that have a positive impact on pupil survival rates or larva survival rates? Because in the human reproductive system, what I know, there's a lot of really suboptimal trade-offs for the mother that exist solely for the benefit of the fetus.
That's a good point.
I just want to drop that. Just continue,
no,
please.
it's a good point. Can't answer it, but yes. Okay. So how does this deal with, what is this connection to humans? So let's look at centenarians. Now centenarians are defined as those people who live to 100 or years, verified to live to 100 years of age or older. The oldest person that ever has lived was a French lady, 122.5 years. And if you look at the number of centenarians, they are rather small. If you were to want to do a global, a global-wide survey of genes involved in diabetes, for example, you could easily pick up the cases and controls of several 100,000 individuals in each case very easily, maybe even 1 million for something like that. If you really were
200,000,
get 4,000 or 5,000 cases, of centenarians. So there are not a lot. Also, 80% of centenarians are actually sick. They're just lucky. Okay. I had a friend who died at 101 with cancer, but he was a centenarian, but he was not healthy. And so there are 20% of centenarians are actually very healthy. They delay the onset, as these two charts show on the top. They delay the onset of cancer on the left-hand side, or cardiovascular disease over here. This is a normative control, and these are the various centenarians of different ages. And so they are coming, they have delayed these things for much longer. The controls started to go down at about age 50 or something, and these guys don't start to go down to about 80 or 90, and even there, they're not very hard. And so the same thing over here. So yeah, so that's one point. They delay the onset, possibly due to the fact that they have less inflammation. Secondly, they age. They have white hair if they have hair at all. There are wrinkles in their skin, but they're pretty active. They may be active and do exercise and walking. There's not one centenarian who can run a race against a 20-year-old and win. That's clear. There are those aging phenomena. Most of them do not have dementia of any kind. So they are living long. They're pretty healthy and all kinds of things. Yeah, their skin is aged and all the rest, but they are healthy. And they are the goal that which would be doing, that we're trying to aim for. And so we think then that let's take all this together and put into the summary slide over here. And up to now, what I've told you is all things that have been they're facts because they've been published in peer-reviewed journals and all that kind of stuff. This slide is a hypothesis. So I propose that our flies and possibly the healthy centenarians live long due to following two major genetic changes. The first is suppressing their immune-based inflammation and thus escaping age-related diseases. All age-related diseases that I'm aware of start off with obesity. Diabetes is not a hormonal problem, it's an obesity problem. And then the effects on hormones come all the way at the end, but it's upstream, it's obesity. Secondly, maintaining the healthy activity of the mitochondria is essential to all organisms. And because most mitochondria by bulk in an organism, especially an older organism, mice, humans, are in fact inactive. They're not doing much. And so there's a loss of metabolic energy. One reason people are frail, not the only reason, but one reason people are frail is that they don't have enough energy to do what has to be done, repair and move them and all the rest. But having healthy and active mitochondria goes a long way to do that. And there are ways to do that. There's a supplement you can take, but no, and so forth. That's another story. Now these are the two main changes. What happens downstream in the fly, as I've described it here, they have a continuous activate energy, movement. They have activation of the R response and so forth. The things I discovered, there's 5 things. That may be different from one species to another. It might be very different in humans than it is downstairs. But the point is, if you have suppressed the immune-based inflammation and maximized the efficiency of your mitochondria, now you're in a position to maintain maintain various kinds of defense mechanisms at a constant level. And you can repair stuff that ordinarily would take a second seat to reproduction. And so that's where I think the connection is. I think that making this, if we could find out how to make those two steps work in people, then that might be the step to get them to almost to die healthy. That's about 110 or so. Okay, let's see. So I think increasing healthy longevity will be difficult, but it's not likely to be impossible. It's analogous to trying to put sand into the top of the hourglass. And that's not too difficult. So thank you for your attention, and we'll take any questions we have.
Yeah.
A little bit of time, I'm sorry. Get me started talking.
Yeah, it should be fine, I think. So I don't know if this must have come up at some point, but I mean, inflammation just seems like it's so detrimental in so many ways, and yet it seems like a vital process. Otherwise, why would we have it? Have you guys thought about the nature of how inflammation, just like why is it around? Like, why does it behave the way that it does if it's going to be such an issue for longevity?
Okay, inflammation does kill the bacteria or whatever it is that's infecting you. Now, as we found out in COVID, of course, it goes crazy and can kill your lungs or whatever part of the tissue as well, which is, of course, not what you want. The evolutionary aspect, there's a case like, for example, in macaque monkeys or baboons, I think macaque monkeys, where the males, when they are under stress, they turn on genes that lead to inflammatory inflammation. And they do that because inflammation also kills. The macaque monkeys are fighting with each other, males, for a female. And so by activating the inflammation, they are getting rid of the bacteria that probably are in their body, having taken from wounds that they got from the other animal. And so they can heal up fast and reproduce again. That they die sooner, that's immaterial. As long as you got genes in somebody else and a baby, you're okay. And I think that's the evolutionary basis for it. And in the female, that same pathway in the females does not activate inflammation. It activates another more protective system.
I might have missed this in your presentation, but did you guys do any tests on the flies to test their ability to withstand disease or something like that versus the LA versus the RA?
We gave them, yeah, they did. And this was it's in the 2018 paper. Both the LA and RAs were treated put in by vials that had bacteria of some sort in them. And their survival was noted. And the ones that the LAs survived much better. Because they downregulates the inflammation, but it's not downregulating the protein molecules that are generated in the flies and the immune system to kill the pathogenic bacteria. So it's not shutting down the whole system, just shutting down the fire, but
right.
not the guns, not the rifles.
Is there any situation for flies that would require them to have inflammation, like that process where it'd be like, oh, these RA flies are more evolutionary fit because they have better inflammation because this particular thing in the wild happens to them?
That's a good point. And the point, we may have the same, you could have the same explanation as in the macaque monkeys. It's known that males will fight each other for a female, and they may get wounded too. Though I know of no data on this, but I know there's data that they fight each other. And in fact, and the males, when they, there's good data that came out from, there's the males, when they are trying to court a female, lose their triglycerides like crazy. And unless they mate, they can die. When they can die from mating because their triglycerides get so low, they're under great stress. And it's possible, does that tie in with inflammation? I don't know, but I think I'd go there and look. You know, the flies are under great stress, and does inflammation then at that point have some beneficial value? It might. I'm not sure.
Okay, any other questions for? There's a few people in the audience who are not, they don't have their video on, but I'm not sure if they have a microphone. Okay. Did you have anything else, Joseph?
Yes. I was just wondering if you've put any work or effort into how what you've discovered can be applied to humans.
I think the human aspect is, I think we want to look at our healthy centenarians. They seem to be the closest ones, and the fact that they apparently seem to have a decreased level of inflammation, and they seem to be fairly healthy. We need better information than with that, but I think that's the closest point to it. And the fact that the flies, all three of these independently derived flies, point to the immune system as being crucial. And I should say this: if you inhibit, there's some data coming out from other labs, and we have some data as well. If you inhibit in any way the ROS enzymes, the enzymes that do defend against oxidative damage, then the animal cannot live long. So that's the first step. But only we made animals in a separate experiment that were very resistant to paraquat, but we only selected for paraquat resistance and not for long life existence. And they could drink paraquat like it was, you know, a bottle of beer or something.
What is
paraquat? Paraquat is a molecule that, when it's ingested, gives rise to all sorts of ROS molecules of various kinds.
I
And
see. Yep.
it's
So they
lethal
could
to
actually survive drinking this literal poison.
yes, they can. And but those flies that we selected for paraquat resistance, ROS resistance, cannot live long.
Oh.
They can drink the damn stuff, but they can't live long. So it's necessary to have
You have to select for the thing you actually want.
upregulation of the enzymes, and that's necessary but not sufficient, because if you just upregulate the enzymes and do nothing else, then the animal doesn't get long-lived either. So it's the first step, but you have to follow through with what I think is a second step, which is efficient mitochondria.
Okay. So we're towards the end of the time. I think that might be a wrap, unless somebody else has another question. Okay. So I'd like to thank Dr. Robert Arking for spending time with us again. And this has been really interesting, and it's pretty cool. They got some really significant results with altering fly lifespan. And this will be on YouTube, so everyone will be able to see it. Okay. I think that's the end.